Hello guys, I'm hoping everyone is doing well, and as I've already promised last few days that I will be posting lectures which concerning different medical issues. I'll be presenting a lot of topics, and as I've already promised you the last 2 months, I said that I'm hoping that I'm expecting I want you to expect that I'll be posting videos concerning different medical conditions. Today, to start with, we will be starting with the mechanism of red blood cell inversion by Plasmodium Falciparum. I think for the past 2 days I've been putting lots of tips concerning how the Plasmodium Falciparum aid the inversion to the red blood cells. So today, first, before I'm starting to present this topic, I want everyone to understand that Plasmodium falciparum is one of the deadly parasitic organism which used to infect human being, and it causes malaria. And malaria is one of the deadly disease which everyone is aware of it. So today I won't go that much in details in each aspect, but I'll try to focus on what I've already told you, that it's a mechanism of this, how this Plasmodium falciparum manage or are able to infect, not only to infect, are able to invade the red blood cells during erythrocytic stage, because we know that Plasmodium falciparum has a lot of stages in its life cycle, but we'll focus only on erythrocytic stage, the merocytic stage, merocytes. How are they what do they have that enable them to invade these red blood cells and affecting the normal blood circulation of the blood, and also cause rosette formations, causing sequestration, and lastly cytoadherence. So to start with, this is diagrammatic features diagrammatic feature of how the life cycle of Plasmodium falciparum is. We see here from the beginning that's once the mosquito, which is a female Anopheles mosquito, bite the human being, we know that the those sporozoites which are released by this female mosquito, Anopheles, they tend to travel and go to the liver. So as they go to mature, and after being matured there, they'll be released as a merozoites to the erythrocyte, to the blood circulation, and infecting or invading this normal RBCs, normal circulatory RBC, which are previous not infected with any infection. So once they are infected, then there are the life cycle continues, and later the gametocytic stage of this Plasmodium will be taken with another mosquito, which is a female Anopheles, and to complete the cycle and to allow the cycle to continue. So this is just a simple diagrammatic presentation of how the life cycle and those stages of this Plasmodium during his life. So first, I want everyone to understand that Plasmodium falciparum is one of the falciparum is a parasitic infection. Parasitic infection, I mean, it is a protozoan. So all protozoans, there are protozoan infection, which there are protozoan, those who are grouped into sporozoites, and Plasmodium is one of those those parasites which grouped in sporozoites. Why sporozoites? These sporozoites these are parasites which lack cilia, lack flagella, and they have a pseudospodium. So these are those which they're not, they can't move from one place to another by using cilia because they don't have cilia, by using flagella because they don't have those. So they have they have their normal system. They have their system which is embedded in between their cell membrane, which is actin and myosin. Actin myosin generative machine, which is embedded between the inner membrane of the parasites and the plasma membrane. So it is between there. This is like a generative machine which it consists of actin myosin, actin filaments, actin myosin complex, which allow them to this machine. Once it is activated, it allows these parasites to move from one point to another, or to invade the red blood cells. The red blood cells. So these sporozoites, which lack those cilia, they are not ciliated, they don't have all those flagella. They are called apicomplexan. Apicomplexan. So all those parasites which lack those I said cilia, which lack I said they lack those they lack flagella, they called apicomplexan. And this apicomplexan diagrammatic feature, it has a anterior polar ring, as we see here at the apical complex. It has a anterior polar ring, intraconoid microtubule, conoid, posterior polar ring, and also it has an inner membrane complex, subpellicular microtubule, and it has apical organelles, and these apical organelles which are found at the apex of this apicomplexan, Rhoptries and micronemes. It also has mitochondria, micropore-dense granules, nucleus, endoplasmic reticulum, Golgi apparatus, apicomplex membrane, and so forth. So this is in general those apicomplexan. They consist of these organelles. But if you go and focus on Plasmodium falciparum, we will see that the structure of Plasmodium falciparum, here we see that it consists of apical prominence, which in front of the apical part, it consists of this Rhoptry. We see there is Rhoptry and micronemes, which these are organelles, secretory organelles, which aid or function or are the one which are very important during the inversion of this Plasmodium falciparum to the infected RBCs. Also, it consists, as I've already said, plasma membrane. Outside of it, it is a merozoite coat. Nucleus, we have seen it. I've already mentioned from the apicomplexan in general. It has dense granules and microtubules. So we see here there is a Rhoptry and a microneme. These are the main things which we'll be discussing much, because they are the one which aid the movement of this Plasmodium falciparum, either in invading these RBCs. So as I said, they consist of micronemes, Rhoptry, plasma membrane, actin myosin motor complex, and inner membrane complex. So between the plasma membrane and the inner membrane complex, there is a generator machine, which is actin myosin motor complex. This is the one which aid movement to this Plasmodium falciparum once they are activated. Also, it consists of dense granules, nucleus, and the mitochondria. As we see, it consists of nucleus. This is nucleus. We'll come to see that it plays a huge role during encoding VAR gene encoding, which once the parasite is inside the red blood cells, we'll see how the nucleus encode the VAR genes, and those VAR genes, how they produce certain proteins which are embedded in the parasitic in that parasitic membrane, and which act as a ligands to the normal RBCs. They act as ligands which are more specific to the normal RBCs receptors or adherent proteins. So we see here during the inversion, I've already explained that there is a micronemes and a Rhoptry at the apical end of this Plasmodium Falciparum, but we see that proteins produced by the apical secretory organelles. What are the proteins which are produced by the apical secretory organelles? And those apical secretory organelles we have already seen that are micronemes and the Rhoptry. So micronemes, they tend to produce Plasmodium Falciparum apical membrane, membrane antigen, which is apical membrane antigen 1, Erythrocyte family antigen, EBA 175, M-trap merozoite specific thrombospondin related anomalous protein, and RPR and CYRPA. So the other secretory organelle is a Rhoptry. This is a Rhoptry. Rhoptry tended to release or to produce Rhoptry effectors. So these Rhoptries, they tend to have certain proteins inside them. So they have Rhoptry effectors. They have the RON complex, which are inside this Rhoptry bulb. So we will come to see that the structure of the Rhoptry and how this Rhoptry aid, how this Rhoptry allow those RONI complex to be embedded into the host cell membrane and allow the apical membrane antigen to tightly bind with the preformed Rhoptinic complex and forming a moving junction, which allow these Rhoptry receptors, which allow these parasites, Plasmodium falciparum, to enter inside the red blood cells. Also, they tended to Rhoptry. They tended to also to secrete protein, which allow to create the parasitovirus vehicle membrane, which act as a protective membrane of this Plasmodium. So during invagination of this Plasmodium falciparum, there is a creation of parasitovirus vacuole by the host membrane. So once it invades or it invaginate inside the host membrane, it takes the membrane of the host membrane and it transform it to make it as a parasitovirus vehicle membrane, which protect this Plasmodium falciparum from normal innate immune response of the host cells. So as I said, these Rhoptry effectors, the other function which they have, they tend to rewire or to make or to rewire the cell membrane of the host. The cell membrane of the host red blood cells, so as to escape from the innate immune system. So from the mechanism, we go direct from the mechanism of red blood cell invasion by Plasmodium falciparum. So during the erythrocytic stage, so here it is in blood. Once these merozoites are released from the liver, so there is a calcium signaling which stimulates micronemes, this microneme fusion with plasma membrane. As we say that this Plasmodium falciparum, they have membranes. They have a plasma membrane, and the inner there's plasma membrane. Then below it there is actin myosin complex. Then inside it there is inner membrane complex. So during calcium signaling, so micronemes, as you said, that these micronemes are at the apical part of this parasitic, this Plasmodium falciparum, at the apical end. So they tend to, it's like they fuse, they facilitate fusion with the plasma membrane at the apical end of the parasite to release protein. So once the fusion occurs, it tends to release those protein which we have already mentioned, which are an antigen, which are apical membrane antigen, which interact with extracellular amylase and bind to the host cell protein. Many microneme protein remain tethered to the parasite plasma membrane following secretion and act as adhesins that allow parasite to bind to the host cells. Some microneme adhesins enable gliding. So these microneme adhesins enable gliding motility by coupling the parasite's actin myosin system to the host cell surface or extracellular matrix. Adhesion complexes are translocated. These adhesion complexes are translocated rearward along the parasite, so rearward, backward along the parasite surface to produce a forward motility. So it's like they tended to they translocated rearward, so along the parasite surface to produce a forward motility. So some micronemes which are produced, some microneme protein which have played part during inversion, EBA 175, RIPR, and CYRP are species-specific microneme protein required for Rhoptry. They are very specific, which are required for the Rhoptry effector secretion, but not parasite motility. So the micronemes tend to produce those apical proteins or micronemal proteins, which aids for the Rhoptry effector secretion but not parasite motility. Other proteins like the microneme-associated CRPMP complex are required for Rhoptry secretion and found across apicomplexa and even in free-living ciliate secretion. So this protein like microneme-associated tend to be found in all apicomplexa, not only they tend even to be found in those ciliated organisms, not only the apicomplexa. After secretion, some Rhoptry effectors, as I said before, they tend to rewire a host signaling to suppress innate immune response, while other effectors, notably the component of Rhoptry-neck. This component of Rhoptry-neck complex, these are the ones which are present, are preformed complexes which are present at the this Rhoptry, which are present in the apical end of the parasite. So these parasites, they tended to move or to live with those RON complex. So once they are with this they are near this during the inversion, they tend to secrete this RON complex and embed them to the host cell membrane, and they will be acting as the receptors for the parasites. These parasites are the one which are releasing these RON complexes, and they tend to these RON complexes, they embed themselves during the invagination. During the process of invagination, they tend to embed themselves to the host cell membrane, and once they are there, they tend to expose a RON complex number 2 to be exposed, so as it can bind with the apical membrane antigen of the to form a tight junction which will facilitate or will act as a moving junction to allow this parasite to move from as I said adhesine complex are translocated rearward along the parasite surface to produce a forward motility. So after secretion, some Rhoptry factor, as I said here, they rewire host signaling to suppress innate immune response, while other factors, notably the component of Rhoptry complex, prepare the site of Rhoptry secretion for apicomplexan or Plasmodium Falciparum inversion. A portion of RON2 becomes surface exposed. So once these RON complexes are embedded to the host cell membrane, there is a portion of RON2 because this RON complexes, it is RON2, RON4, and RON5. These are once they are in a group, they call a RON complex. So this RON2 becomes surface exposed following translocation into the host cell, where it tightly binds to the micronemes protein. So as you said, we have micronemes protein and we have Rhoptry. And Rhoptry is already releasing these complexes. I mean, which complexes? This Rhoptry complexes have been released and they've been embedded to the host cell membrane. Once they've been embedded, so these micronemes proteins microneme proteins such as apical membrane antigen 1, they tend to bind with it. So a portion of RON becomes surface exposed following translocation to host cell, where it tightly binds the microneme protein apical membrane antigen 1. The apical membrane antigen 1 and RON2 complex mediate the tight opposition between host parasite membrane at the site of inversion, hence the moving junction through which the parasite actively invaded the host cell using gliding like motility. So once there is once they form a antigen membrane, apical membrane antigen 1, the complex with RON complex number 2, they tend to mediate the tight junction opposition between the host parasite membrane. How does that happen? Because once this RON complex and AMY1 antigen binds, there is polymerization of the actin polymerization, but we won't go that much in detail. But there are aldolase, which they tend to allow polymerization of actin myosin complex. So that polymerization lead to the movement, lead to the activation of actin myosin complex. So actin myosin complex, it is activated and lead to the gliding like motility. So here is a diagrammatic feature of merozoite ligands. These merozoite ligands are those ligands which are produced by the parasites and are the one which are produced by the micronemes and Rhoptry. So the erythrocyte receptors, they've been shown here. We can see that Rhoptry neck is here, which here is Rhoptry bulb. And then we have a Rhoptry neck, and beside the Rhoptry neck we have a micronemes, which are the one which are trying to produce EBA. And EBA, there is a lot of EBA. We have 175, 140, and 181, and also apical membrane antigen 1, which is also produced with the micronemes, and also we have others which are not of that much significantly important. But we see that there is here at apical membrane antigen, there is bone, it tended to bind with a RON 2 peptide, I mean the RON number 2, or among the complex. So it tend to bind. So this RON complex, first it was not here. It has been produced, it has been preformed from this Rhoptry, this Rhoptry organelle. So it has been formed and it has been released, it has been embedded to the host cell membrane, so as to be forming a receptor for apical membrane antigen. And here we've seen that calcium required for the inversion, and we see there is you see here IBA 175, it is bound with GYPA, and we've seen here a lot of receptors of ligands have been bound to erythrocyte receptors. But not only that, we have to see in details how this RON organelle, how does it work to allow all these features to be to allow all this what I've been explaining. So here we see role of Rhoptry-Microneme protein during inversion. Roles of Rhoptry-Microneme protein during inversion. There is reorientation, organelle secretion, and inversion. So role of Rhoptry-Microneme protein during inversion, it is 3 stages. One is reorientation, then organelle secretion, then lastly it is inversion, when the parasite invade to the normal red blood cell membrane. So here we see during the reorientation, we see here at the apical end of the parasites, we see that there is micronemes, and which inside of it contain apical membrane antigen. And we see here at the orange color, there are RONI complex 2, RONI complex 4, and they've been put in colors. So once you see the red one, is RON 2, and the RON 4, it is a green one, and RON 5, it is the purple one. So in general, once they are put in a group, it is called a RONI complex, and they are preformed. They are previously present to this Rhoptry organelle. And we see micronemes, it consists of apical membrane antigen, and we see the greenish coloration. So once this red color here below it is the host cell membrane. This is the process during inversion. So organelle secretion. This organelle secretion, this Rhoptry-Microneme at the host cell membrane, will come to see, and during the inversion, what happened. We see this Rhoptry complexes, Rhoptry-Microneme complex are released here, and they get embedded to the host cell membrane. How? We see a schematic representation of the apical region during inversion of its target host cell. So we see during reorientation, schematic shows with the electron dense Rhoptry bulb shaded in orange. We see that it has been shaded in orange and at the anterior Rhoptry neck, which it is shown in white here below. It is shown in white. So immediately upon reorientation of the parasite, the Rhoptry-Microneme region contain a preformed RONI protein complex, RON 2, Rhoptry-Microneme 4, and the Rhoptry-Microneme 5 that are inserted into a membranous multilamellar host. The micronemes contain apical membrane antigen 1 are also shown. We see at the B organelle secretion. At the organelle secretion, we see that following microneme secretion of apical membrane antigen is processed and inserted into the plasma membrane of the parasite, thereby providing the ligand. So once they are secreted to the plasma membrane of the parasite, they act as the ligand that bind the preformed RONI protein complex as receptors that are subsequently secreted in association with multilamellar host and inserted into the host cell membrane via a membrane fusion event. So during inversion, once inserted into the host cell membrane, the RONI complex provide the parasite with a parasite-derived receptor that bind to the apical membrane antigen ligand. This receptor-ligand interaction contribute to the tight junction formation as the parasite propel itself into the cell by means of the linear motor. The tight junction translocate along the periphery, so there is activation of that actin myosin generator machine, which act which the tight junction translocate along the periphery of the invading parasite, and hence the parasite will invaginate itself and will stay inside the parasitovirus vehicle, which is a PVM, inside it to reproduce itself and to prepare itself for further pathological pathological events. So here we mark the end of how these parasites, Rhoptry, this how Plasmodium Falciparum invade the red blood cells during erythrocytic stage, during the Merozoite, during the Merozoitic stage. So later we'll have a part 2 of it, which will see what happened inside the erythrocyte once this Plasmodium Falciparum was inside. What happened? How this Plasmodium encode VRR genes, how this Plasmodium secrete or produce different protein to act as ligands against the normal red blood cells. So thank you guys. I think once, please try to subscribe, and I'll try my level best to make sure that I come up with the new presentation of what has to come next. Thank you very much, guys, and have a good day. Please subscribe and even I like it, and I wish you all the best. And if you can comment, please try to comment, and if you have some information to add upon it, try to add upon this presentation, so as we all get we can get something new from different people. Thank you very much, and I'm here, Dr. Mtiko. Thank you very much.